Even though pretty much all animals, except super simple ones like sponges, have a nervous system, ours is probably the most distinctive feature of our species. From writing novels to debating time travel to juggling knives, all of your thoughts and actions and emotions can be boiled down into three principal functions: sensory input, integration, and motor output. Imagine a spider walking onto your bare knee. The sensory receptors on your skin detect those 8 little legs. That information is your sensory input. From there, your nervous system processes that input and decides what should be done about it. That's called integration. Like, should I be all zen about it and just let it walk over me? Or should I not be zen and freak out and run around screaming, "Spider!" Your hand lashing out to remove the spider and maybe your accompanying banshee scream is the motor output, the response that occurs when your nervous system activates certain parts of your body. As you can imagine, it takes a highly integrated system to detect, process, and act on data like this all the time. And when we talk about the nervous system, we're really talking about several levels of organization, starting with two main parts: the central and peripheral nervous systems. The central nervous system is your brain and spinal cord, the main control center. It's what decided to remove the spider and gave the order to your hand. Your peripheral nervous system is composed of all the nerves that branch off from the brain and spine that allow your central nervous system to communicate with the rest of your body. And since its job is communication, your peripheral system is set up to work in both directions. The sensory, or afferent division, is what picks up sensory stimuli, like, hey, there's an arachnid on you, and slings that information to the brain. Your motor, or efferent division, is the part that sends directions from your brain to the muscles and glands, like, hey, hand part, how about you do something about that spider? The motor division also includes the somatic, or voluntary nervous system that rules your skeletal muscle movement, and the autonomic, or involuntary nervous system that keeps your heart beating and your lungs breathing and your stomach churning. And finally, that autonomic system, too, has its own complementary forces. Its sympathetic division mobilizes the body into action and gets it all fired up like, spider! While the parasympathetic division relaxes the body and talks it down, like, it wasn't a black widow or anything, you're fine, breathe. So that's the organization of your nervous system in a nutshell. But no matter what part you're talking about, they're all made up of mainly nervous tissue, which you'll remember is densely packed with cells. Maybe less than 20% of that tissue consists of extracellular space. Everything else, cells. The type of cells you most likely heard of are the neurons, or nerve cells, which respond to stimuli and transmit signals. These cells get all the publicity. They're the ones we're always thanking every time we ace an exam or think up a snappy comeback to an argument. But these wise guys really account for just a small part of your nervous tissue because they are surrounded and protected by gaggles of neuroglia, or glial cells. Once considered just the scaffolding or glue that held neurons together, we now know that our different glial cell types serve many other important functions, and they make up about half of the mass of your brain, outnumbering their neuron colleagues by about 10 to 1. Star-shaped astrocytes are found in your central nervous system and are your most abundant and versatile glial cells. They anchor neurons to their blood supply and govern the exchange of materials between neurons and capillaries. Also in your central nervous system are your protective microglial cells. They're smaller and kind of thorny looking and act as the main source of immune defense against invading microorganisms in the brain and spinal cord. Your ependymal cells line cavities in your brain and spinal cord and create, secrete, and circulate cerebrospinal fluid that fills those cavities and cushions those organs. And finally, your central nervous system's oligodendrocytes wrap around neurons, producing an insulating barrier called the myelin sheath. Now, over in your peripheral nervous system, there are just two kinds of glial cells. Satellite cells do mainly in the peripheral system what astrocyte cells do in the central system. They surround and support neuron cell bodies, while Schwann cells are similar to your oligodendrocytes in that they wrap around axons and make that insulating myelin sheath. So don't sell your glial cells short. They're in the majority cell-wise, but of course when it comes to passing tests and winning arguments, most of the heavy lifting is done by the neurons. And they're not all the same. They're actually highly specialized, coming in all shapes and sizes, from tiny ones in your brain to the ones that run the entire length of your leg. But they do all share three super cool things in common. Number one, they're some of the longest lived cells in your body. There's a lot of debate right now about whether you're actually born with all the neurons you'll ever have, but some research suggests that at least in your brain's cerebral cortex, your neurons will live as long as you do. Cool fact number two, they are irreplaceable. It's a good thing that they have such longevity because your neurons aren't like your constantly renewing skin cells. Most neurons are amitotic, so once they take on their given roles in the nervous system, they lose their ability to divide. So take care of them. And number 3, they have huge appetites. Like a soccer-playing teenager, neurons have a crazy high metabolic rate. They need a steady and abundant supply of glucose and oxygen, and about 25% of the calories that you take in every day are consumed by your brain's activity. Along with all these wonderful qualities, your neurons also share the same basic structure. The soma, or cell body, is the neuron's life support. It's got all the normal cell goodies like a nucleus, DNA, mitochondria, ribosomes, cytoplasm. The bushy branch-like things projecting out from the soma are dendrites. They're the listeners. They pick up messages, news, gossip from other cells and convey that information to the cell body. The neuron's axon, meanwhile, is like the talker. This long extension or fiber can be super short or run a full meter from your spine down to your ankle. We've got a few different axon layouts in our body, but in the most abundant type of neuron, the axons transmit electrical impulses away from the cell body to other cells. For us students of biology, it's a good thing that nerve cells aren't all identical, because their differences in structure are one of the ways that we tell them apart and classify them. The main feature we look at is how many processes extend out from the cell body. A process, in this case, being a projecting part of an organic structure. 99% of all your neurons are multipolar neurons with 3 or more processes sticking out from the soma, including 1 axon and a bunch of dendrites. Bipolar neurons have 2 processes, an axon and a single dendrite, extending from opposite sides of the cell body. They're pretty rare, found only in a few special sensory places like the retina of your eye. Unipolar neurons, on the other hand, have just 1 process and are found mostly in your sensory receptors. So if you ever find yourself probing around someone's nervous tissue, remember these 3 terms to help you figure out what you're looking at. But because we're talking about physiology here as well as anatomy, we have to classify these cells in terms of their function, and that basically comes down to which way an impulse travels through a neuron in relation to the brain and spine. Our sensory, or afferent neurons, pick up messages and transmit impulses from sensory receptors in, say, the skin or internal organs, and send them toward the central nervous system. Most sensory neurons are unipolar. Motor, or efferent neurons, do the opposite. They're mostly multipolar and transmit impulses away from the central nervous system and out to your body's muscles and glands. And then there are interneurons, or association neurons, which live in the central nervous system and transmit impulses between those sensory and motor neurons. Interneurons are the most abundant of your body's neurons and are mostly multipolar. Okay, it's applied knowledge time. Let's review everything we've learned so far in terms of that spider on your knee. Those 8 creeping legs first activate your unipolar sensory neurons in the skin on your knee when they sense something crawling on you. The signal travels up an axon wrapped in Schwann cells and into your spinal cord, where it gets passed on to several multipolar interneurons. Now, some of those interneurons might send a signal straight down a bunch of multipolar neurons to your quadriceps muscle on your thigh, triggering you to kick your leg out before you even know what's going on. Other interneurons will pass that signal to neurons that carry it up your spinal cord to your brain. That's where your body first recognizes that thing as a spider, and the connections between neurons interpret and split the signal so that you can either scream and start swinging your arms wildly about, or remain calm and with dignity remove the spider from your person. It's all based on the connections between neurons, which brings me to a whole new question. How? How in the name of Jean-Martin Charcot do nerve cells use chemistry and electricity to communicate with each other? It's one of the most stupefyingly awesome and complicated aspects of your nervous system and basically of all life, and it is what we will cover in our next lesson.
Let's review some of the key concepts introduced in this video. The peripheral nervous system consists of neurons or nerve cells connecting parts of the body to the spinal cord and brain. There are 2 kinds of peripheral neurons: afferent or incoming neurons, which transmit signals from the body to the spinal cord and the brain, and efferent or outgoing neurons, which transmit signals from the brain and spinal cord to the body. The brain and spinal cord make up the central nervous system, which consists of interneurons, which transmit signals between neurons. Neurons have 4 main parts: the cell body or soma, which contains the cell's nucleus and metabolic machinery; the dendrites, where signals from other neurons are received; the axon, which extends out of the cell body and down which neural impulses are propagated; and the synaptic terminals, which send signals to other neurons, or muscles or glands. Neurons are surrounded by glial cells, or glue cells, which support and nourish them. In the central nervous system, the glial cells are astrocytes, which surround and support neurons, anchor them to the blood supply, and exchange nutrients and waste between neurons and capillaries. Oligodendrocytes, which wrap around neurons, creating myelin sheaths, which are very important to nerve function, and we'll look at them in more detail later. There are also microglia, which provide immune defense, and ependymal cells, which create, secrete, and circulate cerebrospinal fluid. In the peripheral nervous system, the glial cells are satellite cells, which do what astrocytes do in the central nervous system, and Schwann cells, which do what oligodendrocytes do in the central nervous system. There are several kinds of neuronal structures. Some neurons are bipolar, which means that they have two processes extending out of the cell body. Some are unipolar, which means that they have one process extending out of the cell body. And some are multipolar, which means that they have more than two processes extending out of the cell body. These different neurons are found in different parts of the nervous system and have distinctive functions. In addition to these kinds of neurons, there are also pyramidal neurons and anaxonal or anaxonic neurons, which don't have axons. In order to understand how neurons work, we need a little background in physics. Matter is made up of atoms, which are themselves made up of three kinds of subatomic particles. Protons, which have a positive electrical charge, neutrons, which have no electrical charge, and electrons, which have a negative electrical charge. Particles with like charges repel each other, while particles with opposite charges attract each other. Atoms with an equal number of protons and electrons have no overall electrical charge. They are electrically neutral. Atoms with unequal numbers of protons and electrons have an overall electric charge. Atoms with an overall electrical charge are called ions. There are two kinds of ions. If an atom has more protons than electrons, the overall charge of the atom is positive. A positive ion is called a cation. If an atom has more electrons than protons, the overall charge of the atom is negative. A negative ion is called an anion. Ions, like protons and electrons, repel things of like charge and attract things of opposite charge. The force of attraction or repulsion between particles of opposite or like charge is an electrical force. Particles of opposite charges want to come together. Particles of like charge want to move apart. If you prevent them from doing what they want to do, you create electrical potential energy, which is converted into kinetic energy or energy of motion if you let the particles go. These two particles, for example, with opposite charges want to get together. If you keep them apart, you create a tension, a pent-up energy. If you let them go, they move towards each other. The energy of their motion, their kinetic energy, is the potential energy they had before you let them go. And this kinetic energy can be used to do work. These two with like charges want to move apart. If you keep them together, again you create a tension, potential energy. And if you let them go, they move away from each other. Again, the energy of their motion, kinetic energy, is the potential energy they had before you let them go. And again, this kinetic energy can be used to do work. Another fact that's important for neural functioning is that particles in solution, that is, particles floating freely in a liquid medium, tend to move from areas of higher concentration to areas of lower concentration. This process is called diffusion. If you prevent particles in suspension from doing what they want to do, you create chemical potential energy, which is converted into kinetic energy if you let the particles go. So, for example, these guys want to go over here. If you prevent them from doing that, you create potential energy, a tension. And if you let them go, the potential energy is converted into kinetic energy, and again, this kinetic energy can be used to do work. Now let's have a look at how neural impulses are generated and transmitted.
What if everything you did and thought and felt could be communicated by pushing a button? It'd be like using the world's simplest app, one that just sends out a little ping, always at the same volume and length, to communicate everything from "it sure is cold in here" to "I love churros" to "boy, I sure would like to breathe sometime soon." Well, that is actually exactly how your neurons send all the impulses responsible for every one of your actions, thoughts, and emotions. When a neuron is stimulated enough, it fires an electrical impulse that zips down its axon to its neighboring neurons. But they've only got one signal that they can send, and it only transmits at one uniform strength and speed. What they can vary is the frequency or number of pulses, like this is distinct from this. And your brain can translate these signals, reading them like binary code, organizing them by location, sensation, magnitude, and importance, so that you know the difference between "turn up the thermostat" and "oh my gosh, I'm on fire." That buzz, that nerve impulse, is called the action potential. It's one of the most fundamental aspects of anatomy and physiology, and really life in general. It's happening inside of you right now, and we want to make sure that you understand what all that buzz is about. Before we delve into how neurons communicate, we gotta first understand a little bit of our old friend electricity. Basically, think of your body as a sack of batteries. No, I mean, you don't look like a sack of batteries. I'm just saying that your body as a whole is electrically neutral with equal amounts of positive and negative charges floating around, but certain areas are more positively or negatively charged than others. And because opposite charges attract, we need barriers or membranes to keep positive and negative charges separate until we're ready to use the energy that their attraction creates. In other words, we keep them separated to build potential. A battery, just sitting on its own, has both a positive and a negative end, and the potential to release energy, but it doesn't do anything until it's hooked up to a flashlight or a phone or a kid's toy that lets those charges move toward each other, on the way converting electricity into light or sound or children's laughter. In much the same way, each neuron in your body is like its own little battery with its own separated charges. It just needs an event to trigger the action that brings those charges together. If you're thinking that this sounds more like engineering than anatomy, that might not be a bad thing. It might even help to think of your neurons in the same terms an electrician might use. Voltage, for example, is the measure of potential energy generated by separated charges. It's measured in volts, but in the case of your body, we use millivolts because it's a pretty small amount. In a cell, we refer to this difference in charge as the membrane potential. The bigger the difference between the positive and negative areas, the higher the voltage and the larger the potential. And just like there's voltage in your body, there's also current, the flow of electricity from one point to another. The amount of charge in a current is related both to its voltage and its resistance. Resistance is just whatever's getting in the way of the current. Something with a high resistance is an insulator, like plastic, and something with a low resistance is a conductor, like metal. Now, when we talk about these concepts in terms of you, we're typically talking about how currents indicate the flow of positively or negatively charged ions across the resistance of your cell's membranes. And again, these membranes separate the charges, so they're what provide the potential to convert the electricity into something useful. Okay, now that we got Electricity 101 down, let's see how it works inside your nervous system. A resting neuron is like a battery just sitting in that sack that is you. When it's just sitting there, it's more negative on the inside of the cell relative to the extracellular space around it. The difference is known as the neuron's resting membrane potential, and it sits at around negative 70 millivolts. Where do those charges come from? Outside of a resting neuron, there's a bunch of positive sodium ions floating around, just lingering outside the membrane. Inside, the neuron holds potassium ions that are positive as well, but they're mingled with bigger negatively charged proteins. And since there are more sodium ions outside than there are potassium ions inside, the cell's interior has an overall negative charge. When a neuron has a negative membrane potential like this, it is said to be polarized. Now, these ions didn't just show up in this arrangement on their own. This is all orchestrated by one of the most important bits of machinery in your nervous system, the sodium-potassium pump. This little protein straddles the membrane of the neuron, and there are tons of them all along the axon. For every 2 potassium ions it pumps into the cell, it pumps out 3 sodium ions. This creates a difference in the concentration of sodium and potassium, and a difference in charges, making it more positive outside the neuron. This difference is an electrochemical gradient, and you probably know enough about biology by now to know that nature hates gradients. It wants to even out all of those inequalities in concentration and in charge to restore balance. But the only way to even out that gradient is for the ions to pass across the membrane. Thankfully, the sodium-potassium pump isn't the only way in or out of the cell. The membrane is also riddled with ion channels, large proteins that can provide safe passage across the membrane when their respective gates are open. And these gates open and close for different reasons depending on their structure and purpose. Most are voltage-gated channels, which open at certain membrane potentials and close at others. For example, sodium channels in your neurons like to open around negative 55 millivolts. But some others are ligand-gated channels. They only open up when a specific neurotransmitter, like serotonin or a hormone, latches onto it. Then we also have mechanically gated channels, which open in response to physically stretching the membrane. In any case, when the gates do open, ions quickly diffuse across that membrane down their electrochemical gradient, evening out the concentrations and running away from other positively charged ions. This movement of ions is the key to all electrical events in neurons, and thus is the force behind every single thing we think, do, and feel. Of course, not all of your body's electrical responses are the same, and neither are the flows of ions going in and out of your neurons. If only a few channels open and only a bit of sodium enters the cell, that causes just a little change in the membrane potential in a localized part of the cell. This is called a graded potential. But in order to send long-distance signals all the way along an axon, you need a bigger change, one big enough to trigger those voltage-gated channels. That is an action potential. And your best bet for making that happen is to depolarize that resting neuron. I mean, cause a big enough change in its membrane potential that it'll trigger the voltage-gated channels to open. It all starts with your neuron sitting there at resting state. All of the ion channels are closed and the inner voltage is resting at negative 70 millivolts. And then something happens. Some environmental stimulus occurs, say like a spider brushes up against a tiny hair on your knee, triggering those sodium channels to open, increasing the charge inside the membrane. Now, the stimulus and the resulting change have to be strong enough to cross a threshold for the true action potential to kick in, and that threshold is about negative 55 millivolts. Remember that number, because this is an all-or-nothing phenomenon. If the stimulus is too weak and the change doesn't hit that level, it's like a false alarm. The neuron just returns to its resting state. But kind of like Doc Brown hitting 1.21 gigawatts in the DeLorean, once it hits that threshold, you're not going to travel in time, but you are going to see some serious action potential. At that threshold, the voltage-gated sodium channels open, and there are tons of these, so all of the positive sodium ions rush in, making the cell massively depolarized. So much so that it actually goes positive, up to about positive 40 millivolts. This is action potential in action. It's really just a temporary reversal of a membrane potential, a brief depolarization caused by changes in currents. And unlike graded potentials, which are small and localized, an action potential kicks off a biological chain reaction which sends that electrical signal down the axon. Because each of your neurons has lots of voltage-gated sodium channels, so when a few in one area open, that local current is strong enough to change the voltage around them. And that triggers their neighbors, which triggers the voltage around them, and so on down the line. As soon as all that's underway, the process of repolarization kicks in. This time the voltage-gated potassium ion channels open up, letting those potassium ions flow out in an attempt to rebalance the charges. If anything, it goes too far at first, and the membrane briefly goes through hyperpolarization. Its voltage drops to negative 75 or so millivolts before all of the gates close and the sodium-potassium pumps take over and bring things back to their resting level. Now, when part of an axon is in the middle of all this and its ion channels are open, it can't respond to any other stimulus, no matter how strong. This is called the refractory period, and it's there to help prevent signals from traveling in both directions down the axon at once. So that is the surprisingly simple app that your nervous system uses to let you experience the world. And because the voltages in this process are always pretty much the same, the initial threshold around negative 55 millivolts and the peak depolarization at 40 millivolts, your neurons only communicate in a single monotone buzz. It doesn't matter if it's a spider on your knee or an elephant, a paper cut or a stab wound, the strength of that action potential is always the same. What does change is the frequency of the buzz. A weak stimulus tends to trigger less frequent action potentials, and that includes if the stimulus is coming from you, like your brain telling your muscles to perform some task. If I need to do something delicate, like pick up an egg, the signal is low frequency. But a more intense signal, like trying to crush a can, increases the frequency of those action potentials to tell your muscles to contract harder, and the message turns into something that you can't ignore. Action potentials also vary by speed or conduction velocity. They're fastest in pathways that govern things like reflexes, for example, but they're slower in places like your glands, guts, and blood vessels. And the factor that affects a neuron's transmission speed the most is whether there's a myelin sheath on its axon. Axons coated in insulating myelin conduct impulses faster than non-myelinated ones, partly because instead of just triggering one channel at a time in a chain reaction, a current can effectively leap from one gap in the myelin to the next. These little gaps are the delightfully named named nodes of Ranvier, and this kind of propagation is known as saltatory conduction, from the Latin word for leaping. But what happens when an action potential hits the end of its axon and is ready to do more than leap and jump all the way to another neuron? That you will find out next time. Today, you learned how your body is kind of like a big bag of batteries, and how ion channels in your neurons regulate this electrochemistry to create an action potential from resting state to depolarization to repolarization, and a brief bout of hyperpolarization. Thanks for watching, especially to all of our Subbable subscribers who make Crash Course possible for themselves and for everyone else. To find out how you can become a supporter, just go to Subbable.com. This episode was written by Kathleen Yale, the script was edited by Blake T. Festino, and our consultant is Dr. Brandon Jackson. It was directed by Nicholas Jenkins and Michael Aranda, and our graphics team is Thought Cafe.
One more thing before you leave. We like Crash Course a lot, and we hope that you like Crash Course a lot, but I kind of feel like Crash Course is only useful for a certain segment of the population. Like, once you get to a certain age, then it's good, and then forever, it can be helpful to people. But younger people, not so much. And so, we are creating Crash Course Kids, hosted by Sabrina Cruz from Nerdy and Quirky. Crash Course Kids will start out focusing on 5th grade science, but we'll keep expanding to other topics as the channel grows. Sabrina will be talking about food chains and gravity and how the sun works and how plants eat and why flamingos are pink and many other topics. Oh, and another note, teachers, you can rest assured that we've got you covered. There will be info about the standards we've used to make sure that we're doing our very best to help you out. So if you are a teacher, or you know a teacher, or you know a child, or you know someone who has a child, or you've ever seen a child, you can tell them to go to youtube.com/crashcoursekids and subscribe, and you can go do that as well if you would find that kind of content useful or interesting.
Let's review the key concepts from this video. A membrane potential is the difference in charge between the inside and the outside of a cell that results from ions being separated by the cell membrane. The inside of the cell has an overall negative charge due to the presence of negatively charged proteins and negative chloride ions. The outside of the cell has an overall positive charge due to the presence, mostly, of a high concentration of positive potassium ions. Voltage is the measure of the potential energy generated by separated charges. When a neuron is in its resting state, the difference in charge between the inside and outside of the cell is 70 millivolts. That is 70 thousandths of a volt, which is very small. This is called the resting potential of the neuron. In this resting state, the cell membrane is said to be polarized. Actually, the term resting potential is a bit misleading. In fact, the separation of charged particles creates an unstable situation. There is an electrochemical gradient consisting of the overall difference in charge between the inside and outside of the cell, that's the electro part, and the overall difference in concentration of sodium and potassium ions, that's the chemical part. The positive and negative ions are attracted to each other, and there is a tendency toward a balance of the concentration of the potassium and sodium ions through diffusion. Overall, sodium wants in and potassium wants out, but the membrane blocks them. Now, since the resting membrane potential is an unstable state, it will not occur naturally. It has to be made to happen. In the neuron, the mechanism that does this is called the sodium-potassium pump. The sodium-potassium pump is a protein embedded in the cell membrane that pumps sodium ions out and potassium ions in at a ratio of 3 sodium for 2 potassium. The action of the pump creates, maintains, and restores the resting membrane potential of the cell. And let's not forget that an axon is a tube, so sodium-potassium pumps are embedded around as well as along the cell wall. What happens when a neuron fires is that the axon membrane goes from a state of being polarized, the resting potential, to being depolarized. In a neuron, depolarization results in an overall negative charge on the outside of the cell and an overall positive charge on the inside, the opposite of the resting potential. Depolarization happens when there are openings in the cell membrane that allow particles to pass in and out of the cell, restoring electrical and chemical balance. The openings in the cell membrane are caused by the opening of ion channels, which are also proteins embedded in the cell membrane that open and close in response to various kinds of stimuli. These are also embedded both around and along the axonal membrane. There are three kinds of ion channels. Voltage-gated ion channels open and close in response to difference in membrane potential. Ligand-gated channels open and close in response to binding with neurotransmitters or hormones, ligands. And the mechanically gated ion channels open and close in response to stretching of the cell membrane. When the ion channels open in a section of the cell membrane and ions flow in and out of the cell, a new membrane potential, a new difference between inside and outside charge, is established. If this new membrane potential reaches minus 55 millivolts, it's sufficient to open voltage-gated ion channels in the adjacent section of the cell membrane. This membrane potential is called the action potential because it's sufficient to cause changes in other parts of the cell membrane. A neural impulse is an action potential that's propagated down the axon to the synaptic terminals. Depolarization and repolarization of successive segments of the axon membrane results in a flow of negative charge, an electric current. Though this is different from a current flowing down a copper wire, which involves the flow of free electrons. The propagation of the action potential is more like a virtual current, arising from successive depolarization and repolarization of segments of the cell membrane. After the action potential is generated in a segment of the cell membrane, sodium-potassium pumps return it to its resting potential in a process called repolarization. There's a bit of an overshoot in this process, with the cell interior becoming more negative than resting potential at minus 75 millivolts. This process is called hyperpolarization. So a neural firing event consists of a depolarization, repolarization, hyperpolarization, and a return to resting potential of a section of the axonal membrane, which is propagated down the length of the axon. While the ion channels are open, the cell membrane cannot respond to any new stimuli. This is called the refractory period, and it's very short. The sodium-potassium pump helps return the membrane to its resting potential. Now, this process from resting potential to depolarization to action potential to repolarization to hyperpolarization to resting potential happens very quickly, in a matter of thousandths of a second. There's only one kind of signal a neuron can send, the negative charge on the outside of the cell membrane. And this signal can vary only with respect to frequency, how often an action potential is generated, and velocity, how fast the action potential travels. The speed of propagation depends on whether or not the axon is myelinated. A myelin sheath is a non-conducting protein that wraps around segments of the axon, with little gaps in between called the nodes of Ranvier. The insulation causes the signal to jump from node to node, a process called saltatory conduction. In this way, the signal reaches the axon terminals much more quickly than it would if the axon were not myelinated. In an unmyelinated axon, there is continuous conduction, which is relatively slow. In a myelinated axon, there is saltatory or jumping conduction. And notice that after 10 milliseconds, the action potential is much farther along the axon of the myelinated neuron. Now let's have a look at the ways in which neurons communicate with each other.
What's 1,000 times thinner than a piece of paper, more numerous in you than grains of sand on a beach, and proof that the smallest things can sometimes be the most powerful? I'm talking about the synapse, the meeting point between two neurons. If your neurons form the structure of your nervous system, then your synapses, the tiny communication links between them, are what turn that structure into an actual system. Because as great and powerful as your neurons are, when it comes down to it, their strength and their purpose lies in their connections. A single neuron in isolation might as well not exist if it doesn't have someone to listen to or talk to. The word synapse comes from the Greek for to clasp or join. It's basically a junction or a crossroads. When an action potential, and if you don't know what an action potential is, watch the last episode, sends an electrical message to the end of an axon, that message hits a synapse that then translates or converts it into a different type of signal and flings it over to another neuron. These connections are rather amazing feats of bioelectrical engineering, and they are also ridiculously, mind-bogglingly numerous. Consider that the human brain alone has 100 billion neurons, and each of those has 1,000 to 10,000 synapses. So you got somewhere between 100 to 1,000 trillion synapses in your brain. Each one of these hundreds of trillions of synapses is like a tiny computer all of its own, not only capable of running loads of different programs simultaneously, but also able to change and adapt in response to neuron firing patterns and either strengthen or weaken over time depending on how much they're used. Synapses are what allow you to learn and remember. They're also the root of many psychiatric disorders, and they're basically why illicit drugs and addictions to them exist. Pretty much everything in your experience, from euphoria to hunger to desire to fuzziness to confusion to boredom, is communicated by way of these signals sent by your body's own electrochemical messaging system. Hopefully, you know enough about email and texting etiquette to know that if you're going to communicate effectively, you have to respect the sanctity of the group list. It's not a great idea to send a mass text to all of your friends first thing in the morning to give them the urgent news that you just ate a really delicious maple bacon donut. Seriously, people, if you happen to have a friend who truly adores bacon, then an email would suffice. But if you're out clubbing and suddenly Bill Murray shows up and starts doing karaoke, then that would be a totally appropriate time to notify all of your friends at once that something awesome is happening, and they better be a part of it. And in much the same way, kind of the same way, your nerve cells have two main settings for communicating with each other, depending on how fast the news needs to travel. Some of your synapses are electrical. That would be like an immediate group text. Others are chemical synapses. They take more time to be received and read, but they're used more often and are much easier to control, sending signals to only certain recipients. Fortunately, your nervous system has better text etiquette than your mom and knows when each kind is appropriate to use and how to do it. Your super fast electrical synapses send an ion current flowing directly from the cytoplasm of one nerve cell to another through small windows called gap junctions. They're super fast because the signal is never converted from its pure electrical state to any other kind of signal the way it is in a chemical synapse. Instead, one cell and one synapse can trigger thousands of other cells that can all act in synchrony. Something similar happens in the muscle cells of your heart, where speed and team effort between cells is crucial. This seems like a good system, so why aren't all of our synapses electrical? It's largely a matter of control. With such a direct connection between cells, an action potential in one neuron will generate an action potential in the other cells across the synapse. That's great in places like your heart, because you definitely don't want half a heartbeat. But if every synapse in your body activated all of the neurons around it, your nervous system would basically always be in group text mode, with every muscle fiber and bit of organ tissue always being stimulated and then replying all to the whole group, which would stimulate them even more until everyone just got maxed out and exhausted and turned off their phones for good, which would be death. So that would be bad, which is partly why we have chemical synapses. They are much more abundant, but also slower, and they're more precise and selective in what messages they send where. Rather than raw electricity, these synapses use neurotransmitters, or chemical signals that diffuse across a synaptic gap to deliver their message. The main advantage chemical synapses have over electrical ones is that they can effectively convert the signal in steps, from electrical to chemical back to electrical, which allows for different ways to control that impulse. At the synapse, that signal can be modified, amplified, inhibited, or split, either immediately or over longer periods of time. This setup has two principal parts. The cell that's sending the signal is the presynaptic neuron, and it transmits through a knob-like structure called the presynaptic terminal, usually the axon terminal. This terminal holds a whole bunch of tiny synaptic vesicle sacs, each loaded with thousands of molecules of a given neurotransmitter. The receiving cell, meanwhile, is, yes, thankfully, the postsynaptic neuron, and it accepts the neurotransmitters in its receptor region, which is usually on the dendrite, or just on the cell body itself. And these two neurons communicate even though they never actually touch. Instead, there's a tiny gap called a synaptic cleft between them, less than 5 millionths of a centimeter apart. One thing to remember is that messages that travel via chemical synapses are technically not transmitted directly between neurons like they are in electrical synapses. Instead, there's a whole chemical event that involves the release, diffusion, production and reception of neurotransmitters in order to transmit signals. And this all happens in a specific and important chain of events. When an action potential races along the axon of a neuron, activating sodium and potassium channels in a wave, it eventually comes down to the presynaptic terminal and activates the voltage-gated calcium channels there to open and release the calcium into the neuron's cytoplasm. This flow of positively charged calcium ions causes all those tiny synaptic vesicles to fuse with the cell membrane and purge their chemical messengers. And it's these neurotransmitters that act like couriers, diffusing across the synaptic gap and binding to receptor sites on the postsynaptic neuron. So the first neuron has managed to convert the electrical signal into a chemical one, but in order for it to become an action potential again in the receiving neuron, it has to be converted back to electrical. And that happens once a neurotransmitter binds to a receptor, because that's what causes the ion channels to open. And depending on which particular neurotransmitter binds to which receptor, the neuron might either get excited or inhibited. The neurotransmitter tells it what to do. Excitatory neurotransmitters depolarize the postsynaptic neuron by making the inside of it more positive and bringing it closer to its action potential threshold, making it more likely to fire that message onto the next neuron. But an inhibitory neurotransmitter hyperpolarizes the postsynaptic neuron by making the inside more negative, driving its charge down, away from its threshold. So not only does the message not get passed along, it's now even harder to excite that portion of the neuron. Keep in mind here, any region of a single neuron may have hundreds of synapses, each with different inhibitory or excitatory neurotransmitters. So the likelihood of that postsynaptic neuron developing an action potential depends on the sum of all of the excitations and inhibitions in that area. Now we have over 100 different kinds of naturally occurring neurotransmitters in our bodies that serve different functions. They help us move around and keep our vital organs humming along, amp us up, calm us down, make us hungry, sleepy, or more alert, or simply just make us feel good. But neurotransmitters don't stay bonded to their receptors for more than a few milliseconds. After they deliver their message, they just sort of pop back out and then either degrade or get recycled. Some kinds diffuse back across the synapse and are immediately reabsorbed by the sending neuron in a process called reuptake. Others are broken down by enzymes in the synaptic cleft or sent away from the synapse by diffusion. And this mechanism is what many drugs, both legal and illegal, so successfully exploit in order to create their desired effects. These drugs can either excite or inhibit the production, release, and reuptake of neurotransmitters. And sometimes they can simply mimic neurotransmitters, tricking the neuron into thinking it's getting a natural chemical signal when really it's anything but. Take cocaine, for example. Don't take cocaine. Once it hits your bloodstream, it targets three major neurotransmitters: serotonin, dopamine, and norepinephrine. Serotonin is mainly inhibitory and plays an important role in regulating mood, appetite, circadian rhythm, and sleep. Some antidepressants can help stabilize moods by stabilizing serotonin levels. And when you engage in pleasurable activities like hugging a loved one or having sex or eating a really great donut, your brain releases dopamine, which influences emotion and attention, but mostly just makes you feel awesome. Finally, norepinephrine amps you up by triggering your fight or flight response, increasing your heart rate and priming muscles to engage, while an undersupply of the chemical can depress a mood. So in a normal, sober state, you got all these neurotransmitters doing their thing in your body, but once they've delivered their chemical payloads, they're usually diffused right back out across the synapse to be absorbed by the neuron that sent them. But cocaine blocks that reuptake, especially of dopamine, allowing these powerful chemicals to float around and accumulate, making the user feel euphoric for a time, but also paranoid and jittery. And because you have a limited supply of these neurotransmitters, and your body needs time to brew more, flooding your synapses like this eventually depletes your supply, making you feel terrible in a number of ways. Cocaine and other drugs that target neurotransmitters trick the brain, and after prolonged use may eventually cause it to adapt, as all of those synapses remember how great those extra chemicals feel. As a result, you actually start to lose receptors, so it takes even more dopamine and finally cocaine to function normally. Sometimes the best way to understand how your body works is to look at how things can go wrong, and when it comes to your synapses, that, my friends, is what wrong looks like. In their natural healthy state, your synapses know when to excite, when to inhibit, when to use electricity, and when to dispatch the chemical messengers. Basically, a healthy nervous system has the etiquette of electrical messaging down to, well, a science.
Let's review the key concepts of this video. A synapse is the connection between two neurons. There are two kinds of synapses. There are electrical synapses, also called gap junctions, in which the action potential is passed directly from one cell to another. And there are chemical synapses, also called synaptic gaps, in which the electrical signal from a sending neuron is converted into a chemical signal, which is transmitted across the gaps between neurons and is converted back into an electrical signal by the receiving neuron. Communication between neurons via electrical synapses is very fast. Communication via chemical synapses is slower, but the chemical interactions make it possible for the incoming signal to be modified. For example, it can be amplified, or inhibited, or split. Let's look closely at a typical chemical synapse. The neuron sending a chemical signal is called the presynaptic neuron. The neuron receiving the signal is called the postsynaptic neuron. At the end of the axon of the presynaptic neuron are the presynaptic terminals, or synaptic end bulbs. This is one of them. The bulbs are located at the end of a branch of an axon and contain neurotransmitters. The neurotransmitters are held within tiny sacs called synaptic vesicles. A neurotransmitter is one of more than 100 chemicals capable of binding with postsynaptic receptors. On the postsynaptic neuron, there is a region called the receptor region, containing the chemical receptors the neurotransmitters can bind with. The current flowing down the axon of the sending neuron causes voltage-gated calcium channels in the walls of the presynaptic terminals to open. This causes positive calcium ions to flow into the cell and change its overall charge, and this in turn causes the synaptic vesicles to fuse with the cell membrane and release their neurotransmitters into the synaptic cleft, the gap between the sending and receiving neurons. The neurotransmitters diffuse across the synaptic cleft and bind with the receptors embedded in the membrane of the receiving cell. Each kind of neurotransmitter can bind with only one kind of receptor. Neurotransmitters have one of two functions. Excitatory transmitters contribute to the creation of action potentials in the receiving cell by depolarizing its cell membrane, that is, by making the inside of the cell more positive. Inhibitory neurotransmitters contribute to the inhibition of the formation of action potentials in the receiving cell by hyperpolarizing its membrane, that is, making the inside of the cell more negative. This makes it more difficult to excite the receiving cell, since now it takes more excitatory interactions to bring it up to its action potential. After a few milliseconds, the neurotransmitters are released back into the synaptic gap, where they are either reused by the sending neurons in a process called reuptake, or transported into glial cells, or broken down by enzymes, or simply diffuse out of the synapse.